So our next talk today in this area around investment in sort of that next generation of thinking around genetics is Harry Lamb. So Harry grew up just outside Brisbane, and that is really what fueled a passion for agriculture. He commenced a PhD last year in the ag tech genetics field after a background in science and engineering. His project with regards to his PhD hopes to deliver a rapid on-farm genome sequencing tool to the northern beef industry. What he'd like to achieve is playing a role in securing a sustainable future for agriculture and ultimately doing this by promoting adoption of innovative technology by the industry. I think that's a really good fit for what we're trying to talk about today in our overall conference theme around investment in innovation for genetic improvement. So today, Harry's here to talk about crashside genotyping. Please give it up for Harry Lamb.
Cheers, Luke. So thank you very much, MLA, for having me. My name's Harry Lamb, obviously. So I'm from the Agriculture and Food Innovation, based up in Brisbane. And despite my last name, I know very little about the sheep industry. As Luke mentioned, I know a bit about cattle, and so I grew up on a cattle property and have worked on a few stations around Queensland and WA. So Peter did a really good job earlier of summarising the evolution of technology in the last 20 years, and this really is sort of possibly the next evolution of sequencing technology. So the benefits of genomic selection, as you'd all be aware, are firstly that we can predict traits that are difficult to phenotypically measure. And again, Brad had a fantastic example of that with immune competence. Difficult to phenotypically measure, but if we can predict that through genotype, that's a massive benefit. Another benefit is that genotype can be measured from the day of birth. So this means we can take a sample from a calf when it's born and we can predict its temperament for when it's older. So rather than waiting for the animal to grow up and then chasing you around the cattle yards and then deciding you've got to sell it or something like that. So ultimately, this has led to accelerated genetic gain, and this has been very well demonstrated in the dairy industry already. So your traditional snippery pipelines would look something like this, where you muster your herd, you take your hair samples, you then post it off to a lab, and then a couple of days later, your samples will arrive in that lab and genotyping can begin. So once that genotyping begins, our genotype results are ready and uploaded to BreedPlan. And once they're uploaded to BreedPlan, a couple of weeks later, our GBVs are ready for your producers and you can muster your herd and then make a selection decision based on your genomic estimated breeding values rather than just phenotypic inspection. So this is particularly important for the northern beef industry. So the northern beef industry is characterised by very large herds and very large properties. This ultimately means that animals in northern Australia are only mustered. So this ultimately means that animals in northern Australia are only handled or mustered once a year. And for example, when I was in Western Australia, we used 2 or 3 choppers and 5 to 7 motorbikes on the ground to muster our animals. So this costs the owner about $5,000 to $6,000 per day of mustering, and we mustered for a month. So ultimately that means that SNP array genotyping in a lab just isn't feasible for producers in Northern Australia. They can't be expected to bring their animals in, take a sample, send that off, and then have to muster their animals again in 2 months' time. Instead, Northern Australia is still traditionally using phenotypic inspection. So still using your round yard setups or drafting through a race where you're just visually looking at an animal just once and then making a prediction about the value of that animal. That's where my project, crashside genotyping, comes in. So crashside genotyping basically describes the use of a DNA sequencer, which is called the MinINE, to genotype animals on farm while they're in the cattle yards. So what exactly is the MinINE? The MinINE's a DNA and RNA sequencer that's made by Oxford Nanopore Technologies, and it's the only portable and the only real-time DNA sequencer available on the market. So it's about the size of your iPhone, and it plugs straight into the USB port of your laptop or your computer, And it's also comparatively very cheap for sequencing technology. So this costs about $5,000. Other sequencing technologies cost upwards of about $50,000 to $100,000. So the MinION and nanopore sequencing in general uses a nanopore that's embedded in a membrane, and DNA flows through that nanopore and disrupts an ionic current in a characteristic manner. As it disrupts that ionic current, we get what we call a squiggle plot, which you can see up there. And then that squiggle plot is interpreted as a sequence of DNA bases. So when we're sequencing our DNA, we extract the DNA from our sample, and then we fragment it, and then we load it onto the MinION sequencer, and it randomly samples bits of those DNA. Any fragment of DNA that's interpreted produces what we call a read. So that's the interpreted DNA sequence for that particular fragment. So another benefit to the MinION is that there's no theoretical limit to the read lengths that we can produce with this. So in our lab, we're getting read lengths of about an average of about 5,000 base pairs up to 300,000 DNA bases. But there's other reports of 4 or 5 million DNA bases. Unfortunately, though, at the moment, it does have a low individual base accuracy. And that's one of the major challenges that my project is really looking at overcoming. So older technologies that we used to use, they have a much shorter DNA base read length at about 200 DNA bases, but they have a very high individual base accuracy. So the reason this is important is because complex or repetitive regions of our genomes are spread throughout. And these older technologies that we used to use can't accurately characterize those complex repetitive regions. So you can see here in the top, despite not having an error, these shorter reads map ambiguously, or they can map to multiple regions of a reference genome that we have. But the longer read technologies, despite having some errors, they can still map accurately across these repetitive regions. And this goes not only for repetitive regions, but also what we call structural variants or complex larger mutations. And some really great examples of this are the horn phenotype in cattle. So that's controlled by 3 different structural variants. The most popular one is a 212 base pair duplication on chromosome 1. And then in sheep, in European breeds, yellow fat in lambs is controlled by a 7,900 DNA base insertion on chromosome 15. So this MinION technology is actually allowing us to more accurately discover these large structural variants, but also genotype the actual variant and not a SNP marker that is just related to that large structural variant. So basically, at the moment, this is the pipeline we see working for crashside genotyping. Now, bear in mind, like any new technology or anything like that, there's always developments. So we're continuously looking at ways that we can improve each one of these steps and speed them up. So at the moment, we're looking at sample preparation times, and that involves extracting the actual DNA from a tissue sample, a blood sample, or a hair sample. That will take about 15 minutes, and then we have to go into what we call rapid library preparation. So library preparation involves purifying that DNA, and this would usually be done in a lab, but we can actually do it remotely now, and then adding adapters onto each little bit of those DNA fragments. Then we go into our sequencing, and at the moment, we predict we can sort of do enough sequence data for a single animal in about half an hour. Now, if we ran only one animal on each sequencer, it would take forever, but we can actually run multiple animals on a single sequencer. The number of animals we can run at a time, we're not quite sure, and we're still working that out, but that's ultimately going to be where you get the efficiency in terms of time and reducing the cost from. And then we go into what we call bioinformatics, a posterior bioinformatics pipeline. So taking that squiggle plot that I mentioned earlier, interpreting that as genotypes, then doing our GBLUP or SNPLUP and getting your estimated breeding values. So my PhD project aims in particular, the first is to basically see if we can do this. So see if we can get accurate estimated breeding values from low coverage MinION data. And when I say low coverage, what I mean is when we sequence fragments of DNA, that's completely randomly spread throughout the genome. So here, for example, we might have 2 reads that overlap this C position. So we'd say that as 2-fold coverage. Now here we have a 4-fold coverage, but we might have an error in that T base. since we have enough reads there, we can actually still say that that is a G genotype. But where we have lower coverages like this one-fold coverage, we can't determine whether that A is actually an error or whether that A is a mutation in this animal. So that's really what I'm trying to work out overcoming at the moment. So here's some preliminary results, and basically what we've done is I predicted age of puberty in some Brahman heifers, and then I compared different coverages to your snip chip accuracy, assuming that's your golden standard for accuracy. So here you can see I simulated two situations. So one where reference populations and validation populations were genotyped using this, which is simply not going to be the case because we already have our reference populations. But you can see in the validation-only population, you get a much, much higher accuracy at this low coverage. And then as we increase that accuracy, you get right up there with the snip chip accuracy quite quickly. So the next bit is what we call using adaptive sequencing, and using adaptive sequencing ultimately to decrease the cost and increase the accuracy. So adaptive sequencing is this new new algorithm or database that we can use to upload just positions of interest into this software. Then as the DNA is going through this pore, the software decides whether that position of interest is being sequenced or not. And if this particular fragment of DNA is not of interest, it can actually eject the DNA back through the pore, opening it up to only sequence the DNA fragments that we want. So that means that we can easily achieve that 6-fold or 8-fold coverage at just the positions we want. So just at your 50,000 positions that you already have on your SNP arrays, or at structural variants or new variants that Brad might have come out with his immune competence. So my take-home messages are crashside genotyping, we believe in our lab is the future, or something that will look significantly like this, but it's definitely not here yet. It is one of those technologies that will take time. But what it will do is it will make genomic selection more accessible for producers. So producers will be able to genotype their animals when they want on the farm and get their breeding values when they need them while the animals are still in the yards. It will also allow us to add new variants into the pipeline faster. So instead of having to come up with an entire new array, SNP array to genotype your animals when we need a new variant, we can just add a single location into the database that we use to then genotype that variant at no additional cost. It will also hopefully allow for us to incorporate what we call epigenetics or methylation. So signals from the mother's upbringing that are passed down onto your lamb or your calf or something like that, we can then incorporate that into our prediction one day. I just need to thank Liz and Ben Hayes, my supervisors, as well as the Coffee and the Centre for Animal Science, and in particular our whole lab group, and also Meat and Livestock Australia for their scholarship, and everyone here who's invited me, as well as Karis Jones and Angelica Pickup. Thank you very much.
Thanks, Harry. Okay, questions? All right, good. Got a few. So we'll start on the line with a question. We'll take the top one. I think we'll put the second one out of scope there. So how do you see this being applied more at the seed stock level or commercial level?
So the driver... So really for me, the driver for this and my whole project has hinged around the northern beef industry and trying to get breeding values to the northern beef industry so they can use that. But at the end of the day, this technology can be applied anywhere. So at the moment, it probably won't be as accurate as your snip chips. So if you're a stud breeder that's close to town, you're going to keep snip chipping your snip chip genotyping your animals. But in those more remote locations, you're probably going to be better off using this sort of thing where you can easily identify that tail of your herd and remove them quite quickly.
Okay, so we'll take your question here and then we'll go back on the line. So, Brad?
Oh, so great talk, Harry. That was excellent, mate. So I see a real application for this somewhere like feedlot induction for people sending down different finishing paths, because as I mentioned in the talk I did earlier, we're working towards this steer select product, but one of the feedback, things we get from feedlot operators is what's the lag time between when you take your sample and you get a result back. And for backgrounding feedlots, that's okay because they can take it at the start of backgrounding and know where animals are going. But something like this would be excellent at feedlot induction, where a group of animals could come in and you could decide there and then on that day whether you're going to put them down a short fed or a long fed finishing path. So really neat stuff, Aaron.
Yeah, thank you very much. And I think you're right, like there are a whole range of applications for this, and that's a perfect example of how else it could be used.
Really interesting talk. I suppose the two questions I had were, what is the actual time per animal that the machine's going to take, and what would the cost per animal be?
Yeah, so I was waiting for that cost question. No, fair enough, it is. The time, I'll start with the time. At the moment, it's looking like more something that you do overnight. So you bring your animals in the afternoon, you take your samples, you run it overnight, and then in the morning you can draft off. But this technology is like evolving so rapidly that by next year that could be completely different. So to put it in perspective, back in 2014-13 when this came out, you couldn't get enough data from a 48-hour run for a single animal. Now we get enough data in half an hour. So that's, sort of how quickly it's evolving. So hopefully we'll be able to do it in under 10 minutes in terms of how long we run it. Cost, so to run for 96 hours, it costs about $1,200, give or take, $1,500 to $1,200. But we can run multiple animals on a single, what we call flow cell. So theoretically, the very upper limit is 96 animals, but you got to factor in how much data you want for those animals. So the more data you want to be confident, the less animals you can run at a single time.
So, in other words, it's probably not as economically competitive currently as the companies might be, but there's definite logistic advantages.
Yeah, certainly not as economically competitive, but snippery genotyping cost $500 about 10 years ago. So, yeah.
We'll take that one online, Harry. So while DNA analysis is done on the spot, in terms of the analysis as people are used to it, when it's currently done every 2 weeks, do you want to explain the how?
So this question's probably better off for a real quantitative geneticist, but a great example of this is, as I think Hamish mentioned earlier, we were using, up at UQ, we've just done a big project, genotyping and phenotyping 50,000 animals. So from that, we have snippery results or marker effects that we can then use to really rapidly predict traits like fertility we're doing at the moment based off your markers rather than doing a G-block where you take into account the whole genomic architecture of a reference and validation population.
So, Julius, there's no such thing as a free lunch, so we'll put you on the tools. Anything else you want to add to that?
To add to this last answer, well, I think here we talk about predictions from the genotype alone, so animals with no pedigree and all that. Similar to the heifer select they use in Angus, and the ASBV is a lot more, so that's why that takes long. It's not done every day. So Harry, obviously we're talking about something that's going to come, and we can't look at costs and speed now. We have to look at it, what it will in 10 years. But it might be useful to look at the medical world because they're probably ahead of us. Do you know how it is applied there and at what level and speed and that stuff?
Yeah, so that's a really good question. They're actually trying to, well, they've rolled it out for COVID. COVID's, everything comes back to COVID these days, but they're trying to use it to diagnose COVID rapidly in hospitals, but they're also doing it to rapidly diagnose all sorts of different infections when they have no idea about the causative agent. Because you're sequencing the whole genome of whatever bacteria, you're not actually targeting a specific a specific genome or something like that. So that's really the big benefit to whole genome sequencing as opposed to just snippery genotyping, where you're just looking at those 50,000 positions. You have no other information. But yeah, they're using it a lot in hospitals, also vet clinics. I know they did a bit in the US in vet clinics to rapidly diagnose different diseases. And also, people might remember African swine fever. They were using it for African swine fever when that was starting to come through East Timor and through China and stuff like that.
Excellent. Well, Harry, you can now say that your name has some sheep relevance in terms of presenting at a Sheep Genetics Leading Breeder Forum. Give it up for Harry, yours. Thank you.